Hi, I'm Dr. Jayesh Rao, Assistant Professor SKSV College Lanza. In our previous lecture session, we have seen colorimeter, Beer and Lambert's law. Then we have discussed how light decides the characteristics of the test sample. The incident light from the light source can be transmitted, absorbed, or reflected to the sample. The changes occur during the interaction of incident light with the sample reveals the sample characteristics. Incident light can be adjusted to the wavelength of interest with the help of dispersive or non-dispersive elements. This is what we have seen in last lectures. Now today we are going to see the spectrophotometer. The general introduction of spectrophotometer is a photometer is a device for measuring light, and spectro implies the whole range of continuous wavelengths that the light source is capable of producing. The detector in the photometer is generally a photocell in which a sensitive surface receives photons, and a current is generated that is proportional to the intensity of the light beam reaching the surface. A spectrophotometer is an instrument that measures the amount of light absorbed by a sample. Spectrophotometer techniques are mostly used to measure the concentration of solutes in a solution by measuring the amount of the light that is absorbed by the solution in a cuvette placed in the spectrophotometer. The spectrophotometer is useful for measuring the absorption spectrum of a compound, that is the absorption of light by a solution at each wavelength. Scientist Arnold J. Beckman and his colleagues at the National Technologies Laboratory, that is NTL, invented the Beckman-Dewey spectrophotometer in 1940. Now we will see principle of spectrophotometer. The spectrophotometer technique is to measure light intensity as a function of wavelength. A prism or a grating is used to split the incident beam into different wavelengths. It does this by diffracting the light beam into a spectrum of wavelength. By suitable mechanism, waves of specific wavelength can be manipulated to fall on the test solution, detecting the intensities with a charge coupled device and displaying the results as a graph on the detector and then on the display device. So the basic is that each compound absorbs or transmits light over a certain range of wavelength. This measurement can also be used to measure the amount of a known chemical substance. So we should try to understand how does absorption spectroscopy work. Let's consider the absorption of light by matter. This is closely related to quantum mechanics. When particles of the matter absorb light, electrons in the particles are excited. Initially they are in E0 state, that is called ground state, and when they absorb energy in the form of photon or light, they go into E1 or E2 state, that is called excited state. This is the absorption of light. See, this is the particle, light fall on the particle of matter. Initially they were in a E0 state. When they absorb light, they go into excited state. Now, electrons have particular energy levels, and rays of ultraviolet light have the energy to change the energy states of the electron. In relation to quantitative measurements performed with spectroscopy, the consequences of this phenomenon is that there is a large amount of absorption if a large number of target molecules exist in a solution, and only a small amount of absorption if there is only a small number of target molecules. So what spectrophotometer principle shows? Solutes in a solution show characteristics absorption in UV or visible or infrared region of electromagnetic radiation. Characteristics absorption spectrum can be isolated by passing the electromagnetic radiation through a prism monochromator, and degree of absorption of electromagnetic radiation depends on the condensation of solutes in solution. Depending on the range of wavelength of light source, it can be classified into two different types. UV visible spectrophotometer uses light over the ultraviolet range, that is 185 to 400 nanometer, and visible range, that is 400 to 700 nanometer of electromagnetic radiation spectrum. And IR spectrophotometer uses light over the infrared range, that is 700 to 15,000 nanometer of electromagnetic radiation spectrum. In visible spectrophotometry, the absorption or the transmission of a certain substance can be determined by the observed color. For instance, a solution sample that absorbs light over all visible ranges, that is transmits none of visible wavelength, appears black in theory. On the other hand, if all visible wavelengths are transmitted, that is absorb nothing, the solution sample appears white. If a solution sample absorbs red light, that is near about 700 nanometer, it appears green because green is the complementary color of red. Visible spectrophotometers in practice use a prism to narrow down a certain range of wavelengths to filter out other wavelengths, so that the particular beam of light is passed through a solution sample. This is the instrumentation of spectrophotometer, schematic presentation of simple spectrophotometer. The first is light source, then there is from the light source pass through entry slit, then the lens, then prism or a grating, then again the lens, then exit slit, then cuvette, then detector or amplifier, and then readout a digital recorder. Now first we will see the first part, the lamp or the light source. Lamp is a radiant energy source. Provide a sufficient of light which is suitable for marking a measurement. The light source typically yields a high output of polychromatic light over a wide range of the spectrum. For measuring ultraviolet or visible light, two lamps are usually required. One, a tungsten filament lamp, which produces wavelengths in the visible region, and the second is a hydrogen or deuterium lamp, which is suitable for the ultraviolet. Then the entrance slit is passing of polychromatic light. Then the monochromator. Straight beam of light, that is photons, comes from the lens split into several component wavelength. The spectrum selects one wavelength and that wavelength is sent through the sample. The monochromator actually accepts polychromatic input light from a lamp and outputs monochromatic light. The monochromator consists of these three parts: collimating lens, prism or grating, is called dispersion elements, and second collimating lens. Now second is lens, that is collimator, transmits a straight beam of light, that is photon, that pass through a monochromator, that is prism. Then prism is a dispersed polychromatic light from the source into its constituent wavelength by virtue of its ability to reflect the different wavelengths to a different extent. In the prism or grating, that the grating are often used in the monochromators of spectrophotometers operating ultraviolet, visible and infrared regions. Dispersion device is nothing but what? The prism or a grating. Dispersion devices is a special plate with hundreds of parallel grooved lines. The grooved lines act to separate the white light into the visible light spectrum. The more lines, the smaller the wavelength. Resolution, that is after finishing, smaller the wavelength, resolution. Then lens, second type of lens, are collimator. It is a focusing device transmits a straight beam of light or photon that passes through a prism and fall on exit slit. Now, exit slit a wavelength selector, nothing but a wavelength selector. Then cuvette, the monochromatic light from the filter passes through the colored solution placed in a cuvette. You can use either glass or a quartz squeeze, glass material or a quartz material. Generally, for visible light, visible source, glass material can be used, and for UV light source, generally use quartz material because glass material absorbs UV light. Then next part is detector. Convert radiant energy or photons into an electrical signal. The photo cell and photo tube are the simplest photodetectors, producing current proportional to the intensity of the light striking them. It detects the wavelength of light that as passes through the sample. Then the amplifier or display device. It increases the signal so that it is easier to read against the background noise. The data from a detector are displayed by a readout device such as an analog meter, a light beam reflected on a scale, or a digital display or LCD. The output can also be transmitted to a computer or a printer. A spectrophotometer in general consists of two devices, a spectrometer and a photometer. A spectrometer is a device that produces typically dispersed measures light. A photometer indicates the photoelectric detector that measures the intensity of light. A spectrometer, it produces a desired range of wavelength of light. First, a collimator, that is lens, transmits a straight beam of light, that is photons, that passes through a monochromator to split it into several component wavelengths. Then a wavelength selector, that is slit, transmits only the desired wavelength. And the photometer, after the desired range of wavelength of light passes through the solution of a sample in cuvette, the photometer detects the amount of photons that is absorbed and then sends a signal to a galvanometer or a digital display. There are two kinds of optical rearrangements in a spectrophotometer, a single beam or a double beam type of spectrophotometer. Here this is the schematic representation of single beam and double beam spectrophotometer. Here the first, the blank, and in the single beam spectrophotometer, the blank and then the sample must be moved in the beam, adjustments made and reading taken. But in double beam we can keep blank as a reference sample, and at the same time sample can be measured or can be reading taken. Now blank or reference solution need not move. Now we'll see applications of spectrophotometer. Some of the major applications of spectrophotometers are include the following: detection of concentration of substances, detection of impurities, structure elucidation of organic compounds, monitoring dissolved oxygen content in fresh water and marine ecosystems, and characterization of proteins. Detection of functional groups, respiratory gas analysis in hospitals, molecular weight determination of compounds. The visible and UV spectrometer may be used to identify classes of compounds in both the pure state and in biological preparations. Now, what are the advantages of spectrophotometer? It ensures high degree of spectral purity, minimum stray light into the exit beam, that is wavelength outside the desired light is called stray light. Then greater accuracy and more sensitivity, specificity and precision. Thank you.